Hello, everyone, and welcome to the latest installment of OpenEye's mini webinar series. My name is Luke Zimny. I'm a lead scientific support specialist at OpenEye, and I'll be your host today. Our presenter is Gunther Stahl, Application Science Director at OpenEye. Today, Gunther will be talking about SZMAP and GamePlan. Without further ado, I'll hand it over to Gunther.
Thank you very much, Luke. So today I'm going to talk about SZMAP and GamePlan, tools of ours that analyze water to help you design your lead molecules. So what's the idea behind this? It's really about ligand designing informed by water and water thermodynamics. And there are two pieces to this. One, to analyze the binding site and learn what the binding site actually wants. And the other part that is more on the game plan side is the part to create a testable hypothesis. So one part might look like this when you see areas of the active site that are in purple hydrophobic, in yellow hydrophilic, with water molecules, as you can see, filling the pocket up. The testable hypotheses are really more like this when you see areas of water stabilizing a compound and really design suggestions to add one atom, and the green means to add one hydrophobic atom to your molecule to make it more active. This is actually an example I got from a customer. I ran the 2xcs PDB file for them. And when I showed them this one suggestion, there are multiple others, but this one suggestion really got their attention. And I'll tell you towards the end on the reason on why that was. So let's start with SZMAP and the background and how do we treat water in here. If you think about how we could deal computationally with water, there are three stages. Well, one is we ignore it. We run vacuum electrostatics and don't do water at all. I think we're past that. Computational methods are more advanced, so we don't really do that. The next one would be to use water as kind of a continuum solvent approach and use the features of water without actually seeing the individual atoms and the individual water molecules, all the way to the most time-consuming way of calculating water, really putting water molecules, here's the oxygen, so the H2O in your simulation and run a simulation with actual water molecules. Where does our tool fall? Well, it kind of falls right between continuum and discrete water, because what we do is we fill an active site within the protein with a grid, and this grid is filled with continuum solvent. And on each grid point, grid corner, we place a real water molecule, the oxygen on the grid point, and the hydrogens pointing in multiple different directions. So we can calculate the energies that the water experience with the rest of the water in a continuum fashion, and the protein in multiple different orientations. So we can learn about preferred orientations for these water molecules. And we can sample the whole active site by placing the water molecule one at a time at each of those grid points. We do this calculation multiple times. Actually, when there is a ligand, we have to do the same thing. We actually also do this without the protein, so just the ligand, so we can really learn about the differences. What does water experience when it's in the different environments, protein, ligand, and in the complex, just to learn on what's the deal about each of these individual waters. So that's our semi-continuum solvation approach that we use here to learn about the water molecules. Nice thing about this is we can apply this obviously to a whole pocket, everywhere on the active side. It's deterministic. We place the waters just everywhere. We don't care if it's a hydrophobic or a hydrophilic pocket. Things just go everywhere because we can place them there. We don't run MD. We're really just placing the atoms there. That leaves us to, hey, we don't have to do the whole thing if we don't want to. We can do just a subsite. A sub-pocket to explore. We can really use any point in space we want. So we can use the crystallographic water positions to check what our approach says about these waters that the crystallographer placed at some point. Or really any other coordinate is what we would do in terms of creating these hypotheses with one atom in the right placement tells us something about an addition to our small molecule and a design idea we can create as a hypothesis. So that is the sampling we can do anywhere we want. But what does this tell us about these individual water positions? So if we place a water molecule in the pocket and we learn about some energy value for this of 5 kcal per mole or whatever the number might be in the end, we can't really test this anywhere because there's no experiment that lets us take one water molecule out and we learn some energy value. So what we decided to do is we do the whole calculation that we did before with the water molecule another time, so a second time, but with an object that has all the water atoms but no charges. Meaning this is more of a hydrophobic probe versus this one is a hydrophilic probe to check what's going on in the active site. And if we do the difference between those two free energies we get for the water probe and the uncharged object, the uncharged water, we can learn that anywhere where the delta G is negative, the water is actually favored and it's a polar region of the active site. And the color will stay yellow throughout the presentation. So yellow is always polar. And in purple, we will do the non-polar pieces of the active site where the uncharged water wins and is preferred because there is no interaction to be made for the regular water molecules. In reality, it has to go there, but it doesn't want to be there. So really, these are the non-polar areas of the active site. And that can give us insight on the design we want to do on any of those positions in space. So with that little bit of theory in it, let's look at some of the results. What can I expect from a calculation? This is the result of a calculation for the water in the whole active site. So we placed our one water molecule everywhere, and we show you if it's something significant happening to the water. Is it a unusually hydrophilic part? Then you would see a yellow point, yellow water sitting somewhere. Or an unusual hydrophobic part where the unfavored, the non-polar water wins. These would be the purple parts. And from here, we can then take the stabilizing hydrophilic waters and the destabilizing, the places where the waters don't want to be. And if we overlay that with a co-crystallized ligand, that wasn't there for the calculation of these individual positions, we can see that the ligand does interesting things. So it is hydrophobic where it needs to be hydrophobic. It is hydrophilic where we have a clear hydrophilic signal in yellow. Behind this is actually a carbonyl group. You'll see that a little bit better on the next slide. So that carbonyl group really places where a water would feel great and is in a great position. So a hydrogen bond interaction is possible. And our ligand does that. So the ligands tend to make these interesting interactions that waters would do if they are allowed in the active site. So if we, as this is a little confusing, there's a bit much on that screen, but if you reduce this to the most interesting points around this co-crystallized ligand, you can come to the conclusion that there is a very strong color, the deeper the color, the more energy basically to have. There's a strong purple point very close to this methoxy group on the phenyl ring. And there is a yellow area, so a hydrophilic area right off this phenyl ring as well. If you turn that in 3D, you can actually see the directionality, and it really fits well to the direction of this. So what changes could I think about to make to this molecule to utilize these energies that are available so close by? And this is an experiment on a whole series of molecules that Aztecs published a while ago. And what they did in a lot of experiments and a lot of crystallization, as Aztecs usually does, they added a hydroxyl group in this position, which basically points exactly where this water molecule is. So we are replacing this water molecule with a hydroxyl group. Getting something hydrophobic added on to a methoxy is not that easy. The oxygen doesn't allow, so we have to come up with some other substituent that replaces the methoxy. And one position could be, or one idea could be, an isopropyl group, where one of these new methyl groups actually points exactly in this direction where this purple energy, this non-polar energy is to be had. Consequence of these two small changes is 4 orders of magnitude more potent from a 800 micromolar molecule to a 56 nanomolar molecule. 2 changes. If Aztecs only had this tool, we could have pointed them in the right direction, hopefully saved them some time in not making too many analogs. While this is useful, it's still a little bit of, well, you need to understand and look, and can we make this a little bit easier? And that's, yes, we can. That's where these hypotheses come in. Because the 2 strongest single atom additions to this fragment or this molecule that was in the binding site is add 1 atom down here, and here highlighted in yellow. This should be a hydrophilic atom. Well, think about what this could be. And add 1 atom over here to the oxygen. that should be a non-polar. So in this case, it's green, the non-polar and van der Waals only interactions atom is over here. So now with a met chemist or just a comp chemist can think about what can I design, what atom can I put here or here to give me these non-polar or polar features. Hydroxyl might come to mind, maybe an amine or something else that is hydrophilic. And over here, at least from the design, when you look at this, it points to 3 atoms that, okay, replace the oxygen with something else. Isopropyl might be a useful one. And this calculation here, as we are only looking at fewer points, is 10, 20 times faster than the full grid, where we look everywhere on the active site. With this hypothesis calculation, we're focusing ourselves a lot on just this one part of the active site. So useful, I'd say, especially when you think about GamePlan on how does it work. It actually generates 2 different kinds of hypotheses. It analyzes the molecule itself. So it places waters, our pretend waters or our water objects, in exactly the positions where all these heavy atoms are. And it adds substituents. It adds 1 or 2 atoms to it. So if we think about how that will look like for the scaffold analysis, so for the exact molecule, a water molecule in each of these positions where it's green would make van der Waals interactions. A water molecule here, as you've seen before, that is the carbonyl group that I talked about before. A water molecule here would make significant electrostatics interactions indicated by the big yellow sphere. Over here is maybe a potential mismatch. We only need van der Waals. The ligand has a hydroxyl group. Keep an eye on that one. Maybe that's something you want to change to make it better. I think in the end turns out that there's a bridging water down here, so the hydroxyl group is actually needed and good. But at least it points you to interesting positions. Otherwise, the ligand does everything right, and there's not much to change on it as is. So the scaffold is fine. Now let's take a look at these additions, these small substituents that we add onto. On every atom around the molecule, we're adding an atom in sp fashion, so just straight out. In sp2 and sp3 hybridization, so it's a few points around each of those where an additional ligand, an additional atom substituent could go towards. And if it's an sp3 rotor at the end, we will also rotate this thing around. And we do this for 1 atom additions, so chemically intelligent, trying to design with chemistry in mind, and 2 atom additions, and that's the furthest that we go to look outwards on what kind of additions we can do to this. So we are analyzing a few hundred, maybe 1,000 points around the ligand, way less than the full grid for an active site. So instead of 2 hours runtime on my laptop, you are done in something like 10 or maybe 20 minutes, if that long, to really wait for the results to come back. So thinking about this picture again that I showed you before, now we have an explanation for this point, but what about these other ones with these stabilizing or destabilizing waters? So areas where a water molecule will stabilize a complex or be destabilizing the complex here in this purple color. What about those? How do we go about these? And really, that's an interesting question to have. How do we treat water molecules that are around my ligand? There are different strategies, but I need to understand what they are in order to figure out what to do. So one strategy could be leave them alone. Do not disturb. They are already stabilizing your complex. They are helping. Don't mess with it. Or start modifying your ligand to maybe now interact with the water molecules instead of ignoring them. That might help. Use the water molecules as bridging water. Or go all the way and displace the water molecule. Also tricky, and you see a little bit about that in one of my slides here as well. How can we calculate these areas or these interesting areas? Now, I told at the beginning that we're doing 3 calculations. We're running the APO calculation with the grid on the active site. We're running the complex calculation. So wherever there's room for a water molecule in that grid, we will calculate the information and the energy of the waters that are kind of left when the ligand binds. And we can do the ligand alone in the same grid and the same coordination. So we have the information about the position of these water molecules in all 3 cases. And if we combine the energies of apoprotein and ligand, comparing it to the water energies in the complex for these interesting positions, we can see that if the complex wins, this water molecule is a stabilizing water molecule. It helps us in forming the complex. If the other 2 are superior, well, then that's a destabilizing water molecule. And you better do something about that one, because that really hurts your binding. And if you can change that one, good. Please go about it and try. And the strategies you can follow, and we've seen this in one example here. Neuraminidase is an example where you have the DANA, the desoxy N-acetyl neuraminidase that it can bind in here. This is the lower part of the ligand here. It's a 4 micromolar ligand that binds, and there's room for water molecules down here. Actually, in the crystal structure, you only see one of those 2 water molecules. But in our prediction, we see with these purple fields here, a little a destabilizing area. Waters here destabilize the complex. So what can we do about it? The 2 strategies are displace them, kick them out, get rid of them, because that will help me, just not having them there. Or try to improve the interactions with the water molecules, so use them. And both of them were actually done, but the improving interaction is probably not as often thought about. One thing you can do is to change this to an amine down here. It actually is a protonated amine, so it changes charge state. So a lot is happening to that ligand when it's binding as an amide version. But you clearly see now yellow fields down here. So these water molecules now are stabilizing the complex. They want to be here. Actually, you see both of them in the crystal structure now. And it changes from a 4 micromolar to a 40 nanomolar compound. Great improvement. Not all attributed to the water. There's more happening, but at least the water is not hurting anymore. It's not hindering, it is helping. So some part of this can be attributed to the water molecules. Now we have stabilizing water molecules. What to do with those? If we see a stabilizing water molecule, big first recommendation, leave them alone. Don't do anything with them. They are helping, because you might mess things up. But if you want to go after it, there's a big gain you can get. But you really need to replace all the water interactions precisely. Otherwise, you're left with unhappy amino acids. So amino acids that have no partner, now not helping anymore. So really be careful about it. There was a great find in that neuraminidase project where this guanidinium group replaces these 2 water molecules almost perfectly. And that really makes this into a picomolar ligand. Absolutely fantastic. So if that works, great. Otherwise, 40 nanomolar is not bad to begin with. So maybe you just want to leave this area alone. Different strategies, but in order to apply them, you need to understand what's what and what is happening. So let me get back to this fun example before we end this and we get to Q&A. So the topoisomerase example I got here from a customer was really what can we do? How can we design on this one? What can we change? I ran this for the customer. I didn't know anything about topoisomerase. And what I find is that this part up here binds into this DNA binding region. It's all hydrophobic and makes obviously nice interactions here. All of those atoms are doing what they are supposed to do. There's some electrostatic interaction here in the centerpiece, and around down here is all hydrophobic binding to the protein part of this complex in the topoisomerase. Fine. There's not much I can say about that middle part with my water analysis, because that basically is inside water, and putting a water inside water doesn't tell me anything. There's no interactions I can have. So question is how important that middle part really is as a linker between those 2 areas. But now if I start designing and trying to figure out what else is around there, let's look at these stabilizing waters. So areas where a water stabilizes the complex are here on the left, on the right. Up here, there's something destabilizing a little bit deeper into the DNA part up here, okay? And there is a stabilizing water here. Just as an explanation, that stabilizing water is really not in the plane of this ring system. It sits above, closer to above that ring system here. So it's not in the same plane. That will be important when we look at the additions and the hypotheses. But what are the hypotheses that are generated? Well, the hydrophilic hypotheses, and there are a lot of ideas that where you could get more interaction with amino acids, are here about the center part and deeper down here. Many of those touching the areas of stabilizing water, where I thought that, well, maybe not in the first round, be careful with those, leave them alone. So even if we don't use those, there is still a lot of information and a lot of ideas we can follow up on to make changes to this molecule to hopefully make it better. That's the hydrophilic part. If we look at the hydrophobic parts, again, there is some non-polar interaction that points you towards destabilizing water molecules. Better don't touch this area up here. Seems to be more room down here to make a little bit bigger substituent instead of a 5-membered ring, or add an atom and add a methyl group or something to this 5-membered ring. Okay, fine. And then there is this point, or actually these 2 points that point towards the same area of hydrophobicity and hydrophobic attachments that we could use. So that's the one that the customer got so excited about. And why? Well, if we look at this one and point us to this molecule and look at the 2D structure where this potential addition is, the customer knew about a compound that was in the clinic from GSK looking like this. So a very similar compound, actually a little bit bigger down here, something that SZMAP pointed us to there's room to modify something down here, fine. But then additionally, they created a third ring to make this a little more stable. And they were really surprised to see a tool that points them to do something about here, because I think on their own, they didn't think about stabilizing or rigidifying this area with another ring. You could almost think about maybe a ring going this way over here on the right-hand side, as we had these 2 points coming off, but well, it was closing the ring over here to stabilize this molecule in a different way. So that got one of my customers excited about it. I ran many examples for customers. Give me PDB structure, I'll run this. We talk about the results before you get into it yourself. Happy to do this with more folks if you're interested to learn more about how to design with and around water. Please let us know, and I'm happy to answer more questions in the Q&A. Thank you very much for now.
All right, thanks everyone. We do have a couple questions, and we'll kind of try to get to as many as we can in the next few minutes. Gunther, first question: how long do the water calculations take?
Well, that kind of depends really on obviously how big the pocket is that you calculate for. So the volume of the pocket, we're filling it and we're placing the water molecules everywhere. So for a regular SZMAP calculation, I never had a pocket, and some of them were pretty big, that on my laptop with 8 CPUs ran for more than 5 or 6 hours. It scales pretty well, so you can use MPI on as many CPUs as you want. You can bring time down by throwing more CPUs at the problem. The GamePlan calculation, and I think I showed that on one of my slides, they are between 10 and 20 minutes, I guess. That's the most I've seen on my laptop. So hours is the max I have seen on reasonable hardware.
Okay. Let's see here. Next question. Are the atoms added to the GamePlan results real, or are they fake?
Well, yes, they are fake. We're putting dummy atoms there just to illustrate we're trying something. What type of atom really should be there is based on the properties and the features that we put on it in the end. So from that point of view, yes, they are fake atoms. They are dummy atoms. And if you look at the smiles you will see DU in there. So those are really just dummy atoms and not an actual atom, placeholders to indicate interesting areas of the ligand.
Okay.
Good observation.
Okay, next question. Can I use SZMAP to evaluate the stability of crystal waters?
Stability of crystal water. Yes. You can calculate for the crystal waters, because you can feed the atom coordinates of these waters into SZMAP and say, please only try this exact position where the crystal water sits, and then you can do the exact same calculation of apo structure, the water kind of alone in a sea of the water alone with the ligand and in the complex, and you can get stabilization infos for each crystal water. And from the strength or from the amount of energy there, you could also try to relate, get an idea of which of those are more stable or maybe harder to displace and kick out than others. So that calculation is a command line that's actually in the in the examples in our documentation, there is an example for a command line that will do exactly that for you. Go through the water molecules that you want to keep, or the water molecules that are around the ligand once you provide them, and place the probes where you want them.
Okay.
That's a pretty straightforward command line example.
Great. Next question. Does SZMAP and GamePlan, will it take docked molecules or poses as input, like hybrid output, for example?
Sure, yes. Wherever that, it's easy for us to use a crystal structure because, well, that's what we observe. But if you have a docking result, it's a complex ligand and protein, and complexes can be the input into a simulation, into one of these into the SZMAP runs. And absolutely, yes, hybrid docking results. Doesn't have to be from, maybe I shouldn't say that, doesn't have to be from our software. Any complex, wherever you can get it from, you can run through GamePlan and SZMAP to get ideas or suggestions on what to do to the molecules. So yes, docking results work.
Okay. Next question. Can I use GamePlan or SZMAP results for automated design of new chemical matter and have it design new or better molecules?
Yes, it can be the starting point for a more automated flow or workflow. But as you saw, as there was a question, the atoms that we add on to GamePlan, for example, are kind of fake atoms. So you would have to replace them with something more real, with a real atom, and put some features on, some color features. And then you could use something like Brood, our fragment replacement tool for bioisosteric replacement, to put a new group or replace that part of the molecule with something real, a real fragment. So right now that would be a manual process and is not automated, but it can be the starting point for an automated design pipeline to put things together and get things going for an automated design. Question.
Okay. I think we have one more question here.
Okay.
How does SZMAP or GamePlan perform for surface pockets in terms of making informed decisions for ligand design?
Good question. Surface, well, can you call them pockets? Might be the other question, depending on how flat things are. So if you have a ligand actually binding somewhere, even to a relatively flat surface pocket, whenever there's interactions between ligand and protein and the water is not just in bulk, so on the surface exposed side, then you can probably get some information about water and designing the ligand to improve the interaction to the protein. If you're on the surface, on a solvent exposed side of that ligand, those water molecules are no different than water molecules out in bulk. You probably won't learn anything about a water molecule that sits on top of a ligand exposed to other solvent molecules. The interesting part is where the protein is, and there you should get some information because we can figure out interactions from protein and ligand.
Okay. Well, that's all the questions we had, and that's all the time we had. So perfect timing. I think we'll go ahead and wrap things up. Gunther, thank you very much for a great presentation. And just a quick announcement, we'd like you all, if possible, to join us for our next webinar, Thursday, June 22nd. That's going to be with David Hamilton, and he will be doing an introduction to OpenEye applications and the toolkits. So we hope you'll join us for that. We'd like to thank you all for joining us today, and we hope to see you next time. Thanks a lot.
Thanks, Luke. Thanks, everybody.